Hello guys and welcome back to Excel Share Academy. So today we are going to do the second part of site specific recombination. In the previous lecture we have discussed about the transpositional type site specific recombination. So if you remember there are two types site specific recombination SSR. तो एक transpositional हो गया, एक हो गया जो conservative. Okay. So today in this lecture we are going to discuss about the conservative site specific recombination. So it is a type of genetic recombination in which the DNA strand exchange takes between the segments which are possessing at least certain degree of sequence homology. Last time हमने जो देखा था उसमें कुछ भी sequence homology की जरूरत नहीं थी, but in this case it is required. Okay, so for example, the bacteriophage genome getting integrated into the bacterial chromosome. Okay. Now let's see, these are the examples, the structure which is involved in the CSIR. So we have got the pair of the symmetric recombinants recognizing the sequences. Okay. Now these are the regions where the recombination is going to happen. Now the subunits of the recombinants binds these recognition sites and then they undergo for the recombination. So can you see these are the recombination sites and then the recombination is going to recognize these sequences. Okay. Now it is undergoing recognition. So once it goes and binds to these recognition sequences, recombination happens. Okay. Now classification. So again under conservative SSR, there are two types. That is depending on the amino acid present at the active site of the enzyme. Okay, recombinase. That is tyrosine recombinase family and the serine recombinase family. And these recombinase are also known as resolvase and the DNA invertase. Members of the tyrosine recombinase and lambda integrase using ATTP-B recognition sites. So these are the sites which you need to remember, guys. ये आपके exam के लिए है. So where you are going to remember all these sites. Okay. Now coming to this table where it is already mentioned like how the different type of the family members which is coming under the recombinase, that is serine family and the tyrosine family. हर एक family के under एक example दिया हुआ है. Okay. तो salmonella HIN invertase, transposon GN3 and gamma delta resolvase. फिर tyrosine family में आ गया आपका phage, lambda integrase, phage P1 Cre, E. coli, Ser C and Zer D, East FLP. So these are the guys examples. अगर आप पूरे lecture में if you could remember this table, that is also more than sufficient. And I'll just let you know one more thing that in this case, okay, in the case of the recombinase, हम lab में जो सबसे ज़्यादा use करते हैं, ठीक है, lab में for example हमें use करना होता है for the site directed mutagenesis या कुछ भी genetic change लाने के लिए in an organism, then we also use these recombinase. Okay, so mainly in the lab we use Cre जो आपका टायरोसिन फैमिली में बिलोंग होता है. एक एग्जांपल है जो मैं एट द एंड ऑफ दिस लेक्चर आई विल बी एक्सप्लेनिंग. सो स्टे ट्यून्ड एंड लिसन टू द एंटायर चैप्टर. Okay, so let's talk about the tyrosine recombinase family. So during the strand exchange, the DNA cut at fixed points within the crossover region. Okay, and then they are going to make a deoxyribose hydroxyl group. तो जब भी कोई cut होगा तो obviously 3-OH तो free होगा ही ना whenever there is a cut. So these recombinase proteins they form a transient covalent bond to this OH को. तो यहाँ पे आपका एक recombinase ने covalent bond बना लिया. Now when you say transient, it means it is not permanent, obviously, right? Now this phosphodiester bond between the hydroxyl group of the nucleophilic serine or tyrosine residue conserves the energy that was expended in cleaving the DNA. So ये जो bond बना है, that is actually the energy which is being released when the bond was broken in the DNA strand. Now this energy stored in this bond is used for rejoining in the DNA to the corresponding deoxyribose hydroxyl group on the other side. So यानी कि जब recombination हो जाएगा, ठीक है, so ये जो bond है between the recombinants and the 3-OH में जो बना है, that will be broken and जो energy उस time release होगी, that will be used to make the bonds in the DNA strand when the recombination is over. ठीक है. Okay, so let's see with the help of the example also. So as I said, so यहाँ पे आपका एक निक होगा जिसकी वजह से 3- आपका फ्री हो जाएगा, 3- hydroxyl group. To that your recombinase goes and binds. ठीक है. Now what is happening? They are going to exchange the strands. Can you see over here? So 4 recombinants are required over here. So यहाँ पे आपका this strand is going to make the bond, okay, and this strand is going to make bond over here, okay. Now they are going to release. So जैसे ही जो आपका enzyme है and उसका जो bond है with the phosphodiester bond यहाँ पे present है, जैसे ही वो break होगा, so those strands will be rejoined. ये ऐसे ऐसे, correct. So the energy which was stored between the enzyme and the DNA strand, it is used in order to make the bonds between the DNA strand itself. Okay, and eventually all these enzymes are getting released. So guys, this is really very important. Please do understand. अगर आपको नहीं समझ रहा है, just let me know, ping me in the channel, okay, and I'll clear it again. Just follow the diagram carefully. Maybe I can just explain once again. Okay, so there was a nick. Okay, now to that nick, your recombinase comes and binds over there. So you can see that this bond is formed. Can you see here? Bond is formed. So this bond is formed when there was breakage. When there was breakage in the DNA strand. So ये जो strand जब break हुआ, जो energy release हुई, that energy was used to make bond between the enzyme and the phosphate bond. ठीक है? अब क्या होगा? Strand exchange हो रहा है, वहां पे bonding हो रही है. Now you need to release. Okay? Now कैसे होगा? ये बॉन्ड बनाने के लिए आपको एनर्जी चाहिए. That energy they are getting from when this bond with the enzyme is broken. Now अगर आप अभी देखोगे तो ये दो स्ट्रैंड तो हो गए. Now two more strands are remaining, right? So इसको and इसको ligate करने के लिए R2 R4 they are the enzymes which is going to lose the bond formation between these phosphate hydroxyl group, correct? अब क्या होगा? जब ये बॉन्ड ब्रेक होगा तो ये बॉन्ड बनेगा आपका बिटवीन द 5' एंड द 3'. सिमिलरली जब R4 बॉन्ड तोड़ेगा तो 5' OH एंड यहाँ पे आपका बॉन्ड बनेगा. So this is how strand exchange is happening. This is how the recombination is happening in the conservative SSR. Okay. Now let's talk about the serine recombinase family also. So here during cleavage, protein DNA bond is formed similar to the your tyrosine wala via trans-esterification reaction in which the phosphodiester bond is replaced by the phosphoserine bond between the 5' phosphate at the cleavage side and the hydroxy group of the conserved serine residues. Okay. And so look at over here. So you have got the recombinase, जिसका serine group है, उसका OH. Now this OH is gonna attack, this O is going to attack on the phosphate group. So right, it is going to form a bond over here. Now because of which here the transit bond is formed, and what will happen, you can see here is that आपका temporary protein DNA covalent intermediate बना है. After the recombination, then again this goes leaves and you get the DNA strand. ठीक है? So here an OH group form an active site serine is shown to attack the phosphate, thereby introducing a single strand break at the site of the recombination. Okay. Now the liberated OH group on the broken DNA can then re-attack the protein DNA covalent bond to reverse this cleavage reaction. See, अगर आप यहाँ पे देखो cleavage हुआ है, correct? यहाँ पे जो OH है, now after recombination, the another strand का OH आके यहाँ पे उसको attack कर सकता है, okay? And then the DNA is found back. So eventually what had happened कि recombinance आया, उसने bond बनाया, right? Strand तोड़ा, the strand got exchanged, okay, recombined, and then again it comes back, attacks the protein DNA covalent and rejoins. So जितना आपने recombinase में energy लगाया उतना ही आपका वो release भी हो गया है. Now, if you see, this diagram is totally similar to the previous one. So you have got the crossover region, the recombinase recognition sites are there. So the enzymes will come and bind over there. Now again, the attack is there. You can see it is forming a DNA protein covalent bond over here, right? Then you have got this 3' OH group free is there, correct? So after this exchange and all, they can just attack back and rejoin. So what is happening? Eventually it is resulting in the recombination. Okay. So then again, there are two classes of genetic recombination. ये हो गया आपका circular DNA में, ठीक है? So as we said, recombination sites are there. So you're going to have the recombination. अगर आपके पास है यहाँ पे transposable element, so what will happen? This can jump from here to here. ठीक है, ये यहाँ पे जंप होके यहाँ होगा. सो नाउ वी हैव सीन बोथ द क्लासेस ऑफ जेनेटिक रिकॉम्बिनेशन. पहला हमने एक देखा ट्रांसपोजिशनल एंड दूसरा हमने देखा कंजर्वेटिव. ओके, now how this is the example we were talking about, right? Integration of the lambda phage genome into the chromosome of the host cell. So here you can see that phage recombination sites are there, then bacterial recombination sites are there. So they just cross over, and what happens, this results in the integration of the entire phage genome into the host genome. अब क्या होता है, उनकी machinery थोड़ी fast होती है, so that's why they take over the entire host cell machinery and start synthesizing for their own components. Okay, so guys, I hope this is clear to you. Now यहाँ पे एक सवाल आना चाहिए, why does the recombination site is present in bacteria, जबकि बैक्टीरिया को मालूम है कि वो फाज उसको अटैक कर सकता है. अगर हमारे पास ये recombination site similarity होती ही नहीं, then this integration of the phage would have not happened, right? But कैसा है ना guys, see in the evolution, so they have, you know, they are emerging together. अगर कोई बैक्टीरिया अपने आप को resistant कर लेता है with the phage they mutate in such a way that they mutate in such a way that they get similar sequences again with the evolving host cell. तो यहाँ पे क्या हो रहा है, जैसे जैसे आपका बैक्टीरिया इवॉल्व हो रहा है, like if it is trying to become resistant to this particular phage, the phage is also trying to mutate in such a way that it has got the similar sites which can be recognized in the bacterial chromosome. तो वो फिर दोबारा is able and capable of attacking the bacteria and getting its genome inserted into the bacterial genome. Okay, so this is like जैसे जैसे bacteria evolve हो रहा है, the phage is also evolving. Hence, then we have got three types of CSAR recombination: insertion, deletion, and inversion. So बहुत simple है. Insertion अभी हमने देखा in the case of the lambda patch. Deletion में क्या होगा? This particular region will be deleted itself. Inversion A B अगर ऐसा है तो वो invert होके B A में मतलब sequence आपका पूरा change हो जाएगा. So these types of CSAR recombination is happening. Okay. Now let's talk about this study where we are using the these recombinase enzyme. Okay, so if for example in this I'm going to explain about the CRE. Okay, so if you get time you can read about CRE recombinase, and ये सिर्फ LOX P site जो रहता है उसको recognize करता है. Okay, now what are LOX P site? They are the recognition sites. CRE is the recombinase. Okay, now what they have done over here is that we are making a knockout. तो आपने माउस का एम्ब्रियो लिया, you have taken the blastocyst cells, they have taken and grown into the culture dish. Okay, now in these cells what you have done is homologous recombination किया है, neomycin आपने यहाँ पे एंटीबायोटिक डाल दिया. ओके, सो रेसिस्टेंस टुवर्ड्स द नियोमाइसिन. सो इट मींस दैट हम जब भी ये सेल्स को नियोमाइसिन मीडिया में डालेंगे, सो द ओनली सेल्स विच आर रेसिस्टेंट टू दिस, लाइक जिसके पास रिकॉम्बिनेशन हुआ है, ओके, सो इट हैज गॉट द नियोमाइसिन रेसिस्टेंट जीन, देन ओनली दोज पर्टिकुलर सेल्स आर गोना survive. हम those cells will, so we are gonna select those cells only. Okay, now these transformed cells will be then transferred to the foster cells जो actually grey color का है. ठीक है, so this is the foster mother. तो हमने क्या किया, this particular blastocyst we have taken and put it into the foster mother. So आप क्या देख सकते हैं यहाँ पे grey है. Now once it gives the baby, you are going to have the chimeric mouse. So you can see grey, black pouches are, patches are there. Then it is crossed with the normal mouse. Again you get the chimera. Sorry. Then it is again mated with the normal mouse. So now what you get is the heterozygous for gene knockout over here. Normal mouse. Okay, so what does it mean? Now it has got the two copies of the gene, and here the heterozygous condition is there. So this is how you make your transformed or the gene loss variant in the mouse. Okay, एक छोटा सा paper है where they are talking about the coupling the recombinating to Cre-Lox system, which is enabling simplified large-scale genome deletion in Lactobacillus casei. So if you guys have got time, you can go through it to understand, but it's okay. Just remember about the Cre-Lox system. So Lox, आपका recognition sites, Cre is the recombinase. Just remember all these things. That is more than sufficient. So guys, that's all for today, and from next time onwards we'll be starting with the new topic. So तब तक के लिए अपना ख्याल रखिए. Take care.